What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're going to go into DNA replication to get a little bit further in our unit on molecular genetics. So let's kind of dive in and see where we go. Like always, I want to review a couple concepts before we progress on to the new things to make sure your brain is ready and able to receive the new information. So thinking about yesterday with base pairing, what would be the complementary or the strand that goes with this one? So I have a strand of DNA that's T, T, G, A, C, G, A, C. Take a moment, pause the video if you need to, write that down on the sheet of paper, and try to put the base pairs with it appropriately, and then see if your answer is correct here. Hopefully, you actually paused the video and tried it, and you should know that T always bonds with A bonds with T, kind of going back and forth. The A's and T's are always together, and G always bonds with C, and C always with G, so G's and C's are together. So you should have a complement that goes with this, right? T goes with A, T goes with A, G goes with C, A, whoops, typo, that should be a T. Goodness, Mr. Fitz. And then see how I got a couple typos here. I literally just retyped the sequence, so that's a problem overall. I'll get that corrected and send it out to you as well. But the big thing is that it's supposed to pair up, and I just went and straight ahead copied it. Now, A's don't bond with A's, so that doesn't make any sense. So all of this should be corrected. So let's grab a marker here and actually make the corrections like we're supposed to. All right, so this should be a T. I'll write it down below it. Cross all this out, that's incorrect. C's are supposed to bond with G's. G's with C's, A with T's, A with T's, and C's with G's. So mistakes do happen with this. I'm not a perfect person overall, but it is easy to make those mistakes, right? I was doing multiple things at the same time, and I went ahead and just copied it down instead of base pairing it, knowing that rule that A always bonds with T. Did great in the beginning. I don't know what happened there. So let's go ahead and get rid of that erasing. Hopefully your answer was better than mine on the first try, but hey, no one's perfect, right? So let's look at it this way. Now this one I feel a little bit better on because I was doing the math probably while I was doing the other one. So this is kind of using that same base pairing knowledge and using it kind of in a math sense, okay? So instead of saying that A equals T and just writing A's next to T's, we're gonna say the percentages are equal. So if A's always equal T's, that means 14 A's, or percentage 14% A would mean it's 14% T. All right? If I have 25 T's, that means I would have 25% A's as well. So those two go together. Down here at the bottom, I have C's at 8%, so obviously the G's then have to be 8%. So those are all equal. Now remember, how we can find this chunk over here that are the blank chunks is we've got to do a little bit of math. The idea is all the percentages add up to 100. So A's percentage plus T's percentage plus G's percentage plus C's percentage should all equal 100%. All right, so let's kind of work through it here a little bit. Let's start at the bottom and work our way up. So in the bottom, you have 8 plus 8 would be 16. You take 100% minus the 16 that you already have to figure out what these two combined are. So 100 minus 16 is 84. Then you take 84, divide it by 2, because these two have to be equal. And so then you should have 42 for each one. Right? There also is another little pattern here as well. So these all add up to 100%, or you can realize that there's a kind of simplistic view here. Right? If they're supposed to add up to 100%, that means that there should be two 50%, right? If you look here, 8% plus 42% makes it 50%. You do that again over here, 8% plus 42% makes another 50%. Two 50s make 100. So you could make the association of saying, well, instead of doing all that complex math, Mr. Fitz, why don't I just add those two together? And I can get 50, and then I can do it that way. So let's kind of keep the same mindset. Let's do it in a very easy one here. So if I'm doing that same mindset of figuring out what this G is, and instead of doing the whole 100, I have 25 here, so I know I want 50 between these two. So 25 plus 25 gives me 50. And also, that would be the same here. Because this one's an even split. Everybody's the same, right? 4 25s make 100, so that one's an easy one. Let's try the 50 move up here. So if I have 14% here, this one has to add up to 50, right? So 50 minus 14 is actually 36. And if you look over here, so that makes 50%. These two make 50%, therefore it all adds up to 100. So this is kind of an easy thing to do. The goal is getting them to either add up to 100 or knowing that quick association that I just made of these two have to add up to 50, or these two have to add up to 50, or these two have to add up to 50, right? Because it's this kind of idea that, you know, you're trying to add it to 100%. So if T and G equal 50, that means A and C have to equal 50 to balance out. All right? Now, let's go into DNA replication. So this process is a little bit complicated overall, and it does have a lot of steps to it. To replicate something with a double helix structure, you're probably going to have to do a couple things to get there. Now, I do have an animated video that is going to be the next video after mine on the list that you might want to stop and check out as well to see the parts in motion, because it is a little bit of a complicated subject, okay? So look at that DNA replication animation video that I posted as well. It's very short, it's about 3.5 minutes, so that way you can see the little process in action, okay? But I'm going to show it to you through pictures. So it kind of looks like this when you start off with that video. Make sure you check it out. And so DNA replication overall is what's called a semi-conservative process. The prefix semi means half, so it's a half conservative process. What that means is we're going to take DNA and we're going to use the old strands that are there to build new ones off of it. So instead of trying to just literally do side-by-side copying where you have the old thing and you have the new thing together, you're going to break this thing apart so you can read it and then complement it, right? So what semi-conservative means is you have an old strand, the blue ones here in this diagram, mixed with a new strand, the red ones in here, okay? So DNA is always what's called a semi-conservative process. Now, the steps for this is a lot of stuff to take in, so bear with me with all the weird names here. So first things first, to get the DNA to copy, you have to open that double helix up. You can't copy it unless it's exposed. So you need both of those old strands open, and the enzyme that does that is called helicase. And how you can know kind of that it's an enzyme, number one, is you can look at this ASE. Anytime something ends with an ASE, that tells you it's an enzyme, meaning it's a protein that speeds up a reaction. So in here, helicase speeds up the opening of the helix. It literally is a wedge you're going to see in a picture that just kind of breaks through the hydrogen bonds holding the base pairs together and allows the helix to be unwound or opened up. Then we actually have to start, even though we're synthesizing a new set of DNA here or making a replication of DNA, they're going to use what's called RNA to start it off. And the reason they start off with RNA is it kind of serves as a marker, so later on they know where the cell knows where it started the DNA copy, so it can pop that off and kind of fix it as it goes. And so what it uses to do that starting point is called primase, okay? Got that ASE, so it's definitely an enzyme, and it's called primase because it's priming the start. If you've ever done any painting in the house, usually you put a white coat of paint on the wall first, a lot of times called a primer, so it makes the wall a lot easier to cover up, especially if you have dark colors. So you put a white primer on, so that way when you put your new color on, it's a lot easier to get it nice and smooth and even. Okay, so primase starts off the process. So helicase opens it up, primase starts off the base pairing, then you have something called DNA polymerase comes in. DNA polymerase's job is to see, okay, there's an A here, I'm going to put a T next to it. There's a G here, I'm going to put a C next to it. And she's going to keep going until it needs to stop. Okay, so polymerase, see, remember DNA is a polymer. It's a nucleic acid. It's made of a bunch of little pieces called nucleotides. So you're building a polymer. So polymerase enzyme. All right, and then finally, anytime that you do something, you need to check, right? I had an error earlier, so I had to check my error. And that's an enzyme called ligase, okay? There's no easy way to remember this one, unfortunately. Ligase just sees if there's any errors, kind of checks to see if there's any issues. And if there's a base pair that's incorrect, it'll pop it off and put the right one back on. So this will actually look in and see if the fragments are correct and everything is good. And also there is a time where DNA is copied in chunks, so it'll actually kind of close the gaps there and make sure that all the pieces are together. So ligase is a very important job. So helicase unwinds the helix, primase gets it started, polymerase does all the base pairing basically, and then ligase checks for errors. Okay, that's the short, simple, to the point for each one of those. Now, here's the picture of how complicated this thing gets, right? You have the old original strand here that's got to be opened up. That's by helicase here, right? It's wedging in there, breaking it apart. You have primase not shown on here, okay? But you would usually get a little RNA primer at the start, probably way back here. And now DNA polymerase is just base pairing. So this one looks like it's following, right? It's going in the same direction as the helicase here. So we call that the leading strand. I'm going to talk about that in a minute. And then you have one that actually kind of goes in sections and chunks here, and it moves backwards from the breakoff point. So that's called the lagging strand. So DNA polymerase is working on two different strands simultaneously through two different enzymes. All right. So let's look a little bit deeper here and get some other vocabulary. The first one is called the replication fork, this section right here. So this is the spot where helicase really breaks down the helix, breaks apart all these hydrogen bonds, holding everything together, and exposes the DNA strands so that way they can be copied individually. So this has to come through, drive a wedge in between it to allow the base pairs to start connecting. Then we also have two strands that come in. There's the leading strand and the lagging strand. The leading strand is this top section here from the previous diagram that I just zoomed in on. And it's basically just following the wedge. All it does is follow the helicase all the way through. Starts off where helicase basically started and just keeps going along. So it'll just keep going until the helicase stops and then it'll pop off. Okay? And the direction that it goes, and this comes up on test questions quite frequently, is they call it a 5' end to a 3' end. And what that means for you is it's how the phosphates are organized. You don't have to know the exact issues of how they're structured and organized. I don't really care about that. What I do care is that you know that it's always the number 5 to the number 3. That's all you really need to know. If you see a picture that has 5 on one side and 3 on the other, know that it's going that way, right? Or if 5's over here and 3's over here, you know it's going that way, right? So it's always going from the 5 to the 3. Okay, remember 5 towards the 3. Sometimes we call this strand that's being copied here the continuous strand instead of the leading strand because it's always being added on to continuously and it just follows helicase. There's no fragments, there's no breakups involved. All right, the next one is called the lagging strand. Now, if you've ever played video games, lag is where it kind of skips or jumps, right? You have a little bit of a pause or delay in there. So this is where actually the helicase is going this way in that 5 to 3 direction, but this part of DNA, its 5 is on this end and its 3 is down here. So these two strands of DNA, their 5 to 3s are opposite. So this one has to kind of follow the opposite pathway. As this wedge breaks apart, polymerase has to stick on here and then copy its way across, okay? And so this actually creates a lag. So it'll just go fill in until it gets to a full section that's already base paired up for 2 strands. It'll pop off, it'll come back towards this area where the helicase is, it'll connect back on and keep going. So it's this full so it's constantly got some jumps or some skips in it, hence the lag. Okay? It always still goes in the 5' to 3' end. So it's still following the replication rules that it needs to, but it's making it in chunks instead of a nice continuous strand like the leading strand is. All right, so there was a lot of words there. I know it does take a second to get all those right in your head, so feel free to pause it, go back, rewatch as you need to, and incorporate in that video as well that I also referenced earlier. Now, a little bit more information, just a little bit review. So what direction is DNA always copied? Hopefully you said 5 to 3. Remember, 5 to 3, 5 to 3, all right? What enzyme unwinds the DNA? What opens up the DNA? That's called helicase. Helicase unwinds the DNA. Which enzyme is our editor? Ligase is the editor. Ligase is the editor. What does primase do? Primase, hopefully you remember, is the starting point. So it creates an RNA section to serve as that little starter or prime the whole thing and get it started. And then leading strand and lagging strand, well, a leading strand is a nice continuous copy of DNA, right? Moving from 5 to 3, following that helicase and the replication fork. The lagging strand is lagging behind. It's got to pop off and it's got to come back in. It makes it in fragments, still following that 5 to 3 direction. Everything's 5 to 3. If you have any questions, feel free to reach out to me via Canvas. This is a lot of difficult stuff, and make sure you complete your exit slip. That's all I have for today. Have a good one.